Hello, I'm Jarvis, the general leader of this team. Hello, we're Team Atristinct from Shanghai, China. Our team dedicates to design a biosensor using the technique of synthetic biology to construct a functioning plasmid for bacteria, which can make them all the properties we want to provide easier and cheaper ways for farmers to detect the presence of the atrazine in their field. Atrazine is the harmful herbicide, and the current technologies to detect them are expensive and inconvenient. So, it's essential to innovate an alternative to detect atrazine. Therefore, we name our team Atristinct, which means our aim is to detect atrazine and therefore extinct them. To put it in practice, we gathered together in Shanghai and investigate how to practically make a biosensor that can detect atrazine. Through our effort, we managed it in weeks of experiments. Then, we will propagate our products and let it bring more convenience to more people.
Hello, my name is Mirabella. I'm the web team leader. Now I'm going to introduce the background and the goal of our project. Agricultural pollution is the pollution of the environment caused by overusing chemical products. The sources are actually harmful to all the living organisms that obtain energy from the food cultivated on farms. Since the demand for food is increasing around the world, different kinds of herbicide apply to the farms in order to guarantee the yield of crops, making the agricultural pollution more serious nowadays. Atrazine, which is produced by an agrochemical company in Swiss, was first registered for use as an agricultural herbicide in the United States in 1959. It is used by farmers to control broadleaf weeds and grasses that interfere with the growth of crops. Approximately 80 million pounds of this herbicide is used across the US every year, making it become the second most used herbicide in the country. The large amount of usage of atrazine making it a common pollution of soil, drinking water, people, and wildlife. It has been reported that atrazine can induce significant health problems, including prostate cancer. It is also an endocrine disruptor that directly affects the sexual development of amphibians by changing their hormone cycle. Therefore, using atrazine too much will lead to declines of amphibians and many other endangered species throughout the country. However, owing mainly to the superior weeding efficacy and low price of atrazine, the current policy in most countries is reinforcing management instead of explicitly restricting the use of it. Furthermore, methods we currently use for detecting the amount of atrazine are GCMS and LCMS. These detecting methods have evident limitations such as highly cost, not convenient to carry, and hard to operate. Therefore, our goals are to develop a biosensor for atrazine detection using the theory of synthetic biology. It must be simple, convenient, easy to detect, and at the same time possess high sensitivity towards atrazine.
In order to genetically modify the bacteria Pipitida and make it perform the function designed by us, we first extracted the plasmid PME6032 and PBBRMCS2 as vectors, and prepared the LB liquid, a solid medium for cultivation of bacteria. Then, we got a large amount of replication of gene fragment, which includes ATZR, PATZD, GFP, and T7 terminator using PCR. Afterwards, what we did was connecting those gene fragments through overlap PCR and homologous recombination, and eventually put them into vectors to get functioning plasmid PME6032 ATZR and PBBR1 PATZD GFP. During this process, we had to keep utilizing argon gel electrophoresis to test whether we had got the expected DNA. Restriction enzyme and ligase were also frequently used to clip and or connect gene fragments in order to construct the plasmid we wanted. After that, we got the peptide, which can theoretically perform a specific function. Therefore, our last step was to test if those bacteria really work. Some of our teammates have also attended the final function test. We carry out the test by putting CYA into genetically modified peptide and check whether they will produce green fluorescent protein, which will emit green fluorescent light under specific condition.
We did PCR to obtain ATZR, PATZD, GFP, and T7 terminator genes. The PCR reaction system and procedure are shown in this slide, and the PCR result is shown in the picture below right. We also did overlap PCR to get PATZD, GFP, and T7 terminator genes connected. The PCR reaction system and procedure are shown in these three slides, and the PCR result is shown in the picture below right. PME6032 plasmid and ATZR gene were digested with two endonucleases, BAMH1 and EcoR1. PBBR1 and CS2 plasmid and PATZD GFP T7 terminator gene were also digested with two endonucleases, KPM1 and XHO1. These products were recycled and ligated using T4 DNA ligase. The reaction systems are shown here. PBBR1 and CS2 construction is ligated twice. The PME6032 construction and PBBR1 and CS2 construction were transformed into E. coli DH5 alpha competent cell. Then microbial PCR was done to further identify and sent to my biotechnology company to be sequenced. The purpose of the project is to detect ultrazine in soil and water. CYA is the degradation of ultrazine. Then we did functional test experiments to test the efficiency of detecting CYA. The constructed plasmid were transformed into Pseudomonas petida. After reading related articles, we choose 0, 50, and 100 micromole as the detection concentrations of CYA. We sampled at 0, 1, 2, 4, 6 hours after CYA were added into the conical flasks, and 2 technique repetitions were sampled. KT2440 is a Pseudomonas petida with no plasmid transformed, and KT2440 B and D is Pseudomonas petida that co-transformed with PME6032, ATZR and PBBR1 and CS2, PATZD GFP T7 terminator plasmids. After preliminary analysis, we found that the reaction has no specificity when the concentration of CYA is 100 micromole. So the GFP values at 100 micromole CYA should not be taken into consideration when the Pseudomonas petida is not co-transformed under different concentrations of CYA. The GFP fluorescence value does not change significantly along with time. However, when plasmids B and D are co-transformed, the GFP fluorescence value increases along with time when the concentration of CYA is 50 micromole. This can be explained as follows. As time goes on, more and more ATZR is transcribed. PATZD is activated when ATZR combines CYA, leading to more transcription of GFP. When GFP is lighted by exciting light, there is green light emitting out.
Human practice is also very important to our project. During the process of our research, we did multiple activities and make sure our project is closely related to the society.
I'm going to talk about the interview. We have interviewed with Mr. Liu, who is the Deputy of National People's Congress, Due to the large area of cultivated land in the city, atrazine becomes the most popular pesticide. But it has negative effect on the crop cultivated in the future because it has long residual period in the soil, which will reduce the production. We also learn about the government's management measures. The expert group, they will review the task record and to avoid the farmers' excessive use of atrazine. As a result, nowadays the detection method of government is mainly the combination of inspection and random sampling. So we need to find a more convenient way to detect it.
Hi, this is Rick Chen, and I'm responsible for the section of the survey. In order to get to know the public's knowledge about atrazine and its harm, we posted a questionnaire on the internet and we received 606 responses. The results were shown in these figures. The first investigation is about people's concern about safety of the environment. From the figure, up to 98.6% of the people who interviewed are concerned about safety of the environment. However, most of them didn't hear of atrazine as a herbicide. In the next figures, we can get the conclusion that people who think atrazine remains in crops and water sources and eventually ingested by the body. We also agree with this perception that atrazine is harmful to the health and the environment. In these figures, we can see the distribution of the acceptance of the view that we need a more convenient and easier operation detection technology is roughly skewed to the left, where most people share strong agreement. Thus, we know that a more rapid operation detector is being expected to be used. In Figure 8, the distribution of the government management shows that it is roughly skewed to the right. We regret to know that our survey also gives us public's option towards environment problems related to atrazine and the importance of our research.
Hi there, this is Edith and I'm responsible for the section of public engagement. According to our previous investigation, we believe that it's very important for people to know the harm of atrazine and control the use of it. As a result, Joy Lab spent a lot of time to advertise our project in diverse ways to arouse the awareness of the general public. Firstly, we wrote 6 tweets and uploaded them to WeChat official account to inform more audiences about our project. Second, we shot and edited several videos about atrazine. After that, we also uploaded them to Bilibili to appeal to more audiences. Not only that, but we also designed a banner and a brochure that include the abstract of our project, offering our audiences useful and helpful information. Finally, we went to a stadium for an offline lecture. During the lecture, we introduced the negative effects caused by the overuse of atrazine. A lot of enthusiastic people were interested in the project and asked for more information. That's all what we did for public engagement.
Hello everyone, my name is Arthur. I'm going to talk about the section on fundraising. To have a better and deeper engagement with the public and give our research for more funding, we also conducted several activities to raise money. We made some derivatives such as pencils, bags, tags, and mugs. These products are printed with our team's logo and some unique patterns. We also established an online shop to sell our derivatives. To promote our derivatives, we made some posters and video to advertise. With the help of posters and videos, a lot of people got to know our derivatives. In the end, we thought we can earn 500 yuan. We sold these derivatives in various ways. But for some reasons, some of the derivatives stagnated in our warehouse, and finally we earned about 300 yuan. Finally, we decided to distribute the rest of the inventories to local communities through non-profit events to increase our awareness. Funding population activities is also a very good opportunity to exercise our sales ability.
According to results from our survey, since our research has a potential for business, we also did some marketing analysis. We used 2 methods to evaluate our product. The first method is called the pestle analysis. In the pestle analysis, we analyzed political, economic, sociocultural, technological, environmental, and legal factors that are associated with our product. This helped us know the general environment outside and the threats and opportunities for our product. The second method is called SWOT analysis. In this analysis, we analyze our product's strengths, weaknesses, opportunities, and threats to our product. At last, we concluded that the advantages of our product are fast and cheap. We can cooperate with some big companies and get good resources. We need to pay a lot of attention to the market and the other product. Also, we will keep seeking how we can improve our product and devote to improving life quality for all. With the continuing collaboration with others and our firm determination, We are sure that our product will make our world better. We generated a business plan which can be found in our wiki website if you want to know more detailed information.
In conclusion, by doing all these activities, we effectively arouse the awareness of the general public, understand our program and product profoundly, and develop the commercial value of our product as well.
Now let's talk about the collaboration part. We believe it is important that we learn from each team and share information that will contribute to both teams during the collaboration. We met some IGM teams, for example, phosphate, the filters, and refills. We have learned a lot from them during the collaboration. First of all, expert consultant. It is important to keep the information authoritative and updated, so we must ask the right and essential questions when we are interviewing the experts. Cooperation with wet lab. We should make sure that the information and progress from both labs are updated, as they will keep the both labs at the same pace. For the preparation of the merchandise, it is important to find a balance between the profit and the budget. Overall, collaboration helps us to exchange ideas that we learn from our own experience and also the information that more experienced teams give us.
The results of the experiment have proven that by co-expressing plasmid B and D, Pseudomonas putida KT2440 can effectively detect the presence of CYA within certain concentrations. It is reported that atrazine can be degraded into CYA via ATZA, ATZB, and ATZC enzyme, and the detection of atrazine degradation can also be done. We did not do any experiments about this since time was limited during the training. Therefore, our future plan is to design primers to construct new vectors for atrazine degradation test and atrazine biosensor functional test in order to confirm the practicability in reality. We hope that we can transform atrazine into CYA and then directly detect the GFP intensity using the biosensor we have already constructed. We are also curious about an intriguing question. Can this biosensor detect a small dose of atrazine pollution in water? Thus, we are willing to conduct further research to examine the detection range of our plasmids, aiming to find out the upper and lower limits, as well as determine special environmental factors that may affect the functions. Our subject is undoubtedly novel and promising. We will continue our research to create our final product, living bacteria kit that can be manufactured.
Hi everyone, this is Rain, and I'm responsible for the illustration of DrawLab's future plan. Our plan includes regularly updating WeChat tweets, making more billable videos for further promotion, and continue to sell our peripheral products in our online shop. And those updating might include the progress of our experiments, more introduction about the products, or some interesting facts or tips about the use of atrazine or other pesticides. Besides, our team won't be able to launch the products on the market, mainly through the microblog, WeChat, Zhihu, and Bilibili, as our main idea is to improve existing detection methods and build public awareness of this affordable and convenient way to test atrazine, and further try to minimize the harmful effects of atrazine on the environment and our society. So that's all about our future plan.
Now, let me sum up the contribution of the DRY team. At the beginning of the activity, the DRY team member released an online questionnaire to understand public understanding of the research object and their needs, which provided general direction for the DRY team's research. At the same time, we also uploaded the tweets and video on the online platform, such as the WeChat official account and Bilibili for project promotion. We also completed the production of the wiki, then through the sale of the product around, fundraising and other ways to obtain the activity fund. At the end of the activity, the member of the team presented a brochure, distributed the brochures to residents in the community, and introduced the content and purpose of our activity to the general public. Finally, I would like to thank team members for their hard work in the library in front of the computer. I would like to thank Professor Liu and Professor Wang for their willingness to spare precious time to answer our question. I would like to thank everyone for their hard work.